because he is the main in charge. And one more thing, just which system is coming. Like, you know, there are.
If the record symbol is gone, STBY will come. Okay,
so
that time you can call.
like the project wise,
Sure.
that is going to be recorded.
The record is gone.
For example,
STBY,
like
the symbol
for the
green color is
one,
there.
RF one, there is
Stand
coming
there.
three of them.
Just to call me.
Yes, so for those, just keep it as a RF, and you can use one of them. And other two you can keep for like one AMR, one RF, and one for genomics, which you are going to only use that. So that anything related to the surveillance, that will be one use. And one you can use for the AMR, 80 microbial resistance one. Then one possibly we will keep it for like if anybody wants, then we can give them. Okay, meanwhile, and then just make a catalog for yourself. So that finally you will monitor and you need to also get the what we call as ID. I will just try to add your name also to that. So because the system will come, then after that you need to get the access. I'll just give the information to Chandran also, but you also connect with him. Okay? Yeah. Yes, I think Papri only Papri you so can one more looking for the other four systems, correct? Yes, four system is coming from the DSP system is coming. So he should be also in the loop. Just ask once Papri that which funding it is coming. The main one or one just clarify. And if it comes from the then keep Shashi also in the loop. Shashi in the loop. If it is coming from the what you call welcome plus one, then keep Anurag. And also Shraddha, I forgot to keep Shraddha for the rocket one. Just note it down, and after that you can just send it across, correct? Yeah, and keep Vikas Sani and Suresh in the loop because they use the main in charge for the that group will also work. Okay. Thank you so much. Okay, all right. Take care. Okay, how are you guys? I was asking something else. Are we happy? Yes? Very good. Are you confident of using ggplot too?
No.
Very good.
Honestly.
I need honest opinion on it. So let's revise. Everybody did this one, na? Age scholar, gender scholar. So I hope everybody is here. Just run from the beginning of the code. Use RM, okay? This thing should be there in the top. So that if there is any error, then we will get to know. How are you, baby? Not good. What happened? Tired? What is not coming? Go there. Where is the code first? Can you just find out where is the code for this one and this one? Tell me. Very good. You want to change what color? What is the color right now? Violet. Okay. So do one thing. Just run from this one. Don't think. ggplot. Only from ggplot. So if you assign to a variable, this is what, correct? You can run. temporarily also, like this. It is saying there is ggplot is not found. It means you have to load this one first. So you have to find first error. Okay, this error is coming now. First you have to fix the error. Then only you should work. It's fine? Is it okay? No problem. It was the error was there. You should look at the error. Correct? Now it is saying that theme pub is not there. It means you have to call the library. What is the library? Baba. Okay. Okay. Now. Now it's saying that there is plus
library
then first we have to first run this one, because you have to assign first. There is a some computer. Then you can you should have asked me, na? I was there only, next to you. You should have called me, come na, help me. Now I have given my number, na? You should call me, na? Call me and ask. Okay, so I have shared my number, correct? Feel free to ask me whenever you feel problem. You got the yesterday material for your chemistry?
Yes, sir.
Very good. Read and let me know. I will give you more information. Okay. Like, can you just turn off the light? Very good. Huh? Video? It's okay. I'm not fair on you. It will come like that. Okay. Everybody is done? Are you able to execute the code? Yes. No. How many no? How many no? Dinata? Dinata? Doing? Okay, first you have to create the age scholar, correct? If you create age scholar, here, run this one first. Again you are doing RM, na? Delete this one. If you do this, then it will get created here. Yes. Perfect. Come down. Run again. Yes. Now run. Very good. No, it's fine. Figure is fine. But you are not running this one, na? Okay, so you are putting the what do you call this, your figure outside in the PDF, so it's displayed in the video. If you run separately this one, then it will come here. Okay, understood? Very good. Is there butter?
Sir, some error is coming.
What error is coming? Run one by one. Where is your library? Call them. There is the power. Call library. Library. Iko na. Library. Library. Digi.2. Yours?
Sir, I can use separately on this one.
So your PB is not there. Yeah, you will able to view that. Now run.
No code.
No code. Which code also it's one. Yes, where is the problem? Go out. Yeah. No. Now do move control 2, and type P _BP. Yes, we have enter. There is there. And when come down, come down. You have to some grid, na? Just run the grid. Yes, whenever you restart, you have to run again.
Enter, na?
Yes.
Only when you are releasing up, you have
So if you remove the device off, okay, this is what the phone. This is fine. Everybody is here. Very good. Take the video for the all the screens. So beautiful pictures. The screen only. Different colorful. Okay. So you guys did not understood that much, correct? So we will just do a recap again. Okay. Just make a new file 12 dash ggplot recap. Let's make a very small data frame. Toy df data dot frame let's type CT only, CTCA. Control N case. Is it fine? Okay. So for this, what is the visualization we can use? Many cases. Suppose I want to see what is the expression values of group and the sex wise. Okay. If your value is like group is, suppose we keep as a x, and if sex is a y, then how there the expression is related, for example. So we can use very important what you call as data which we call as a heat map, just to highlight that how these two categorical values can be represented. For example, to do that, first what we will do, library ggplot2. Okay, please run this so that our we have our package. Is it done? Okay. Now same ggplot2 we call data equal to toy df. Correct? Then what we mention about AES. AES for mapping the data. Okay, you can write mapping equal to. So I'll just put in after one after another so that it becomes easy to remember. Okay, mapping is x equal to what? Let's suppose we keep our group. Okay. Then comma y equal to sex. Is it done? Is it done?
Yes.
Very good. Now as we know, after each function this we need to give what? Plus, because we want to add something more information. Then enter geom underscore tile. Okay. So we will make styles. Then AES fill equal to, what was the variable name? Our expression expr. Are you able to get the same figure? So what is the problem with the figure right now? Can anyone suggest? So you see in our data, if you look closely, we have how many values? Total? 6. How many we are displaying here? 4. So what's the problem? What is the problem we have right now? That we are only doing group CA and CT, then male and female. Okay, where the values are summarized. For example, female CA, how many values are there? Female CA, there are 2 values are there. Correct? So the 2 values are summarized. Likewise, if you can see CT is there and female, how many are there? 2. That is also summarized. Male CT, there is only 1 values are there, which is close to 1. Okay, that is fine. Male CA, male then CA, that is around, the value is 4. That is also almost same. Correct? Is it fine? So to reduce this confusion, what we will do, we will create something called sample information. Sample is equal to C of S1 comma S2 comma S3 comma S4 comma S5 then S6. Okay. So I'll just run this again, and now I will place in place of group, I will just say x equal to samples. So now you have the correct visualization. Okay. So I'll just take out the code for you so that you can do first. Yeah. So first is the sample, just include that sample in the column, first column. Are you able to make the figures? Not yet, no? First type this. First do this, then copy this again and change in the group sample. Okay? So you will have first figure is this, second figure is this. Just copy this code and just replace your group information into sample. Go serial, no? One up. Okay, this sample S1, S2, S3. You are just giving the sample information. So for those who have done, they can use additionally the scale information. Yesterday we talked about scale field underscore gradient. There is a call gradient 2 is there. I will just try gradient 2. So you will have the your color code change into very differently. You can try spectral also. For example, distiller you can also try. Distiller will be this. Then you will have Regu identity. Okay, this is better. So you can add color for yourself also. Okay. You can try the low and high, value, everything you can change. Gradient and you can give the values directly. And this here is the sequence gradient. Is it done? So one thing you see with the figure, this particular block is coming very it's not detectable, correct? This looks like a white space. So in this case, what you can do, you can add the boundary called color is equal to suppose black. Okay. Go to the tile. You see the difference? Now you have the box made here. Try this one color. In the tiles, you have to just after the fill, take out this and call equal to black. Color equal to black, it will give the boundaries. Okay. Is it done?
Yes, sir.
Who is not done yet? So can anybody explain me what is heat map now? So in the x-axis and y-axis you have categorical variable, okay, and inside the boxes, the tiles, what you have? The intensity corresponding to the value, continuous value which you want to keep. Okay. So what is the prerequisites for making heat map? You should have two categorical variable and one continuous variable, correct? Is it fine? Is it fine? Is it fine? Okay, very good. And what is the format of the data it should be? Let's discuss that one again. Just do view toy data. So what data type we call this one? Long format. What is we call as long format? Okay. Is it done? What is white format then? Can anybody? What is white format? Anyone? Okay, now let's discuss about how to convert your white format to long format. So first install one package which is called reshape. And one is reshape 2. Okay, there are two packages that you have to install. One is reshape and one is reshape 2. I think you have already installed. You don't have to install. You install already. Reshape and reshape 2. Okay. Focus on the screen. So what we have actually talked about, there are two types of format is there. One is we call as a wide format and one is long format data frame. Okay. So typically now if you look at the wide data has a column for each variable. Okay, what? Wide data will have for each variable you will have the column. For example, ozone, wind and temperature here. Then this is a long data format where you have number of IDs are there, then the variable are kept in a variable column. Okay, and you have the only the values. So this is called what? This is a long format. What is the difference between this? In the wide format you have the variable name column as the one, then you have the values corresponding. Correct? For example, your age, then student name, that will be what? It's a wide format, not long format. Okay. If you have all the variables in one column, which is like ozone, then you have the values corresponding value, then wind, then the corresponding value, temperature, then the corresponding value, then we call this as a long format data. Okay. Why this is useful, we will come below. And what are the things that we can do? So reshape 2, you can convert your wide format data into long format data. Okay, and the long format data also you can convert back into wide format data. So that's why it's called as reshape. Okay, you can change the shape of the data depending on your requirement. So what are the two functionalities that they have? This is a melt where you take wide format data, then you convert into a long format data. Okay, and in the cast what you do, you take long format data, then you can cast into a wide format data. Okay, so now let's come with the example here. So there is a data already there which is called as air quality. So I will share this what you call as material to the WhatsApp group so that you have the access again. Okay, so the material is there. So let's now come to your here in the R Studio. So first what we will do, we will do data. First load the library reshape 2. reshape, I think reshape only we require, na? Reshape 2, yeah, reshape 2 you require, reshape 2. Then data, air quality, load the data, call air quality. Load the data is air quality. Is it fine? Is it done?
Yes.
Okay. So you see in the names typically the column names, call names of air quality. The column name of what is the first letter is in capital letter. So what this is function is doing to lower letter, it is converting your any uppercase to lower, call names of air quality. So it is converting into a lower case. Why it is converting into lower case? So that we can do easily the transactions. Okay. So now what I will do, I will just change my column names of ARQ. Okay, let's first do one thing. Let's keep a very short name called AQ. Okay, for air quality. Is it fine? Okay, it will be easy for us to manipulate. We don't have to type everything. So column name of AQ is this. Now we will convert into a two lower call names of AQ. So what we have done is that we have converted our call names to small letter. See, this was first it was in the capital letter. Everything is now is lowercase. Okay. So that is the function to lower. For doing upper it is called to upper. Okay. Call names AQ. So it will convert all the letters into uppercase. Okay. Just remember this one. Is it fine? Is it done? So now AQ. Okay. So if you do the head, it is like it will just print the top portions, head of AQ. It will show you the top values like 1, 2, 3, 4, 5, 6. 6 rows it will show. Head of AQ if you do, this is the values. Okay, right now what is the format this one? What is the format? This is white format. Why? Because the variable name is in the top. Okay, and the corresponding values are written like this. We will just convert our, so you can type AQ. AQ is in white format. Okay. Now we will just convert into long format AQ L, then melt AQ, then after that you can do again health AQ long. So you should get variable and the values.
L.
Is it done? Is it done? Just do the head and check whether you are getting the desired output. You should have the ozone and hold the variables in the one column for variable, then the values. Okay? Okay? Is it done? Is it done? Is it done? Very good. Okay. Let's work for him. Wait for him. And use the head of AQS. AQS. Yes. And over there. That AQS. Small. Melt AQ. Hey, power. Here. Put it down. चांस नहीं मिलता है कि लाइक हरी what is this? You're writing the code, taking the photo, then writing.
Sure, start up, sir, up here.
Is it done? Okay, very good. Now, very interesting that we will do. Okay, this is little bit advanced, so I want everybody to concentrate here. So here we are saying something else, that you convert into long format but keep month and day intact. Okay, we'll see how it looks like. So you have the same AQL like this, the variable in the value. So what is the difference? Month and day are repeated, okay, for each of the variable. So how the AQL data looks like, let's see, so that you will appreciate. So for example, here the month and day, that column is retained, but only the transformation is done in where? These 4 parameters. Okay, why this is important? We will see that. So what it is showing, for every given month, for every day, you have captured air quality data of ozone, solar radiation, wind and temperature. Okay, now if I am converting blindly, you convert everything into a long format, it does not make sense. Why? Month and day are not variables, they are covariables, covariates. Correct? For example, you take gender as a covariate, but you have captured some more clinical parameters. The clinical parameters can be similarly like ozone, solar, wind and day. and your month and day is the time point that you are capturing. Correct? For example, this month, this day. So if you want to ask, like for example, across the month, okay, what was the ozone level differences, you will not able to ask from this data, long format data. Why? Because all the variables are inside one column. Correct? But now you can ask from this one, this wide format. You can ask the question that based on the different months, what is the distribution of ozone? Okay. And second thing, with this one, what you can ask is that given for every month and with the variables, show me the distribution. For example, here the variables are what? Ozone, then you have solar radiation. So we'll do one exercise first with the AQ data, just this one, then we will move to the MD. Okay. So first what we will do, we will do the box plot between month and the ozone. Is it fine? Okay, let's do that. So what we will do, GG plot. Okay, and you have AQ, comma, AES. What was X for us? X is the month. Okay, month. Can you just concentrate whatever I am typing there so that you remember the thoughts? Okay. So AQ is the data, and what visualization want to do? We want to see the distribution of ozone across different months. Okay, that is our question that we are asking. For that question, the best representation is a box plot because it will show you the distribution. Then what will be there in the Y? Let's suppose ozone. Okay, plus geom box plot. So you see there is a problem with this. 6, 7, 8 is a numeric variable. Correct? So since we are asking this numeric variable should be factor. Factor means it should be categorical. First we have to convert that one. So how we will do that? So you can directly say in here S dot factor. So it will convert your numerical data into factor. Okay. You see, so the differences. So if you don't convert your data, for example, if I just say AQ dollar, what was the month? This is in numeric, correct? So if we don't convert this, then we will get figure like this. So what was the issue? We have to first make into a categorical variable, which is a factor here. So now you have labels 5, 6, 7, 8, 9. Okay. So that's when we did the S dot factor. It is automatically creating our visualization effectively. Is it fine? Okay. Now let's suppose you want to create this. Okay, for all the variables. It is very difficult with the current data of AQ. But with AQ MD, you can now create them. Why? Because you can say in your what do you call as the values correspondingly. So let me give you one more hint. GG plot. There is AQ underscore MD, AQL MD, comma AES X equal to S dot factor of the month. Okay, month. Then you have Y. Y is what here? Y will be what for AQLMD? The value, the corresponding value. Okay, because that's the numeric value. And plus, if I do geom box plot, it will show the distribution of the months and all the values. But what we need, we require for each of the variable. So one of the very best thing is that in ggplot2, for example, I can color my variables as field equal to variable. Now you can see all the parameters, 4 parameters together in each month. You see the beauty? When you want to visualize all the parameters in a single, figure. Okay. So how we did this? So there are 3 mechanisms are there. So first is that whether your data is in right format. So for this, using this one, you can create individual ones. For example, I can create for solar radiation, then wind and temperature. Okay, I can create 4 different figures using this format. But if I actually transform this into a very specific, question specific, data, which is the long format here, you can say you give me the month wise, the corresponding values, then you color them with the variable. That's it. So you will get the specific for each month, the distribution of all the variables. Is it fine? Okay. Now second thing, I will just make it little bit more beautiful, so that because each of the variable has a different unit. If we are keeping like this, it does not make sense, correct? It is okay, but we have to make sure that each of the variables are in a separate panel, let's suppose. Small sub panel want to make. So there is a very nice function available in ggplot2, which is called as plus facet. Okay, facet. Suppose we say wrap facet, the name is variable. Now you see, ozone is very nicely represented, solar is also temperature, wind, all are segregated. But still we are not able to say that units are totally different, but now the x-axis limit is minimum of the value and the maximum of the value. Okay, how you fix this? So there is a way to fix this also. You can say in the wrap facet that the scale here you can do fix, currently is in fix, but we want to free the y-axis. Okay, what happened? Free y. Now you see all the values, ozone variables are in its own scale, then solar scale is in separate, temperature is totally separate, and wind speed is totally different. Is it fine? So now you learn that what is the purpose of having a long format and a wide format. With wide format we will get restrictions. With long format you can play modularly. Okay, you write for one, you can use that one for all. Okay, is it fine? Okay, let's now make. Okay, now start typing. So first this one with the wide format, this is ozone. Okay, and this is for all. I hope you guys are writing this one, correct?
Sir, MD1.
Okay, this one?
Yes.
Okay. So only id.var you have to mention which variable you want to retain. Okay? This is really useful, for example, when you will be doing for genomics. Suppose, for example, for microbiome you want to do the diversity. You want to plot xenon diversity, alpha diversity, beta diversity, and all everything you want to plot in a single figure. This is what it is, you can do that. Okay?
not found in that month. After running this AQL MD. This is long format one and this is white.
आईडी क्या लिखा है तुमने? अच्छे से देखो आईडी क्या लिखा है?
ID values not found in the data month.
नो तुम्हारा कैपिटल लेटर में क्यों लिखा है मंथ में? स्मॉल में हाँ कंप्लीट करो।
ये ग्राफ में तो सिर्फ
एक पहले ये एग्जीक्यूट करो, एक ख़त्म करो पहले ज़िंदगी में, फिर उसके बाद आगे बढ़ो। हाँ नेक्स्ट।
ये ग्राफ में तो सिर्फ मंथ को ही रखा है हम लोग।
क्या मंथ को लिखा है?
मतलब एक एक्सिस में मंथ को रखा है।
हाँ एक्सिस में मंथ को लिखा है यस।
डे को कंपेयर नहीं कर रहे।
देखो हम लोग क्वेश्चन ही पूछ नहीं रहे ना, वो क्वेश्चन ऐसा पूछोगे तो फिर डे के हिसाब से भी कर सकते।
Hello?
What?
This is fine. Is it done, beta? It is only G plot you take. Without no. Go up full time. From top, yes. From the put here. Continue from the most contact. Change your holding. So you will yes. Okay. Why is the problem? Because you cannot convert your policies into money. Make it easy. Now why you guys are not putting your photo easy here. Easy. Because of the plus control minus right plus. You increase your font size so that you can work effectively, please. Why you have separated your track?
Actually, this is whole version.
Whole version. Plus 2. Okay, is it done? Come for a tea. Come for a tea break.
Anima.
Those who got your passport photo, please give me, okay? This one month date. Done. This one. The month day also done. You run it. I cannot do this. Hey, you didn't even type MP here. That's right. This is different.
Okay. But sometimes it's not working. It won't work. You have to press here.
to what file you are using?
And he can use it.
Oh, that thing you have to convert into biopsy, no?
That we have to take two
But
files.
Sandeep also showed that is not necessary as it is.
It's good to have sir. It's good to get
The base. Activate Honda activate
Okay, first thing I think we will do the installation for two packages. One is we have to first install our next flow. Okay, so next flow. I'm just sending you the link for this in the WhatsApp group. So what are the requirements we have? Very good. Java command. Okay everyone, so first to install the next flow you require the Java. Okay, so first you have to install the Java or you can also try, there is one more option is there, you can install the next flow using the Bioconda. So if you do this, conda install -c Bioconda next flow. Okay. So before we start doing this, we will first try to do couple of stuff, which is for Massimo we need to install the next flow. Correct? You remember the what you call as that? Let's get the name. I forgot. Next flow. We have. What is the Massimo? Anybody remember? GitHub?
GitHub Maestro. SI.
Yeah.
On track.
So here you have ON track 2, correct? This was we have to install. So it says here, if you look it closely, so this is next flow minion based pipeline to track species biodiversity. So on2 is a next flow implementation of ONT track pipeline, a rapid, accurate, minion based barcoding. So this is the your original software. Okay, if you click here, so this was made for, it's a rapid and accurate median based barcoding pipeline for tracking species and biodiversity on site. Okay, starting from median sequencing reads, the ON track pipeline will able to accurately consensus sequences in 15 minutes per sample on a standard laptop. Okay, moreover a pre-processing pipeline is provided so to make the whole bioinformatics analysis from raw fast files to consensus sequence straightforward and simple. Okay, so what are the prerequisites? For example, it requires Miniconda, which we already installed. Everybody is familiar with this one, correct? Then it's what is doing, it is just taking your ONT GEMP CPU version interest.tr, which you can actually download from this one. Okay, GIMPI installations. Then you require this particular specific, which is your NCBI. Okay, I'll just show you the demonstration. So there are two ways of doing this. One without the next flow and one with next flow. Without the next flow is what? ONT track. Okay, ONT track. Then with the next flow it is ONT track 2. Okay, so let's do this. This particular part we will not do, but what we will try to do, we will just install this particular Conda environment. Okay, so to do that, let's start with without the next flow. Okay, so this part is done. We need to actually install the Guppy. Okay, we require the email address for this one. Anybody is having this software? Anybody? Okay, Massimo provided this software. Let me register.
We are having our own password, nanoport. What password is there? No password. Have you operated in our system?
No.
Oh no. That day you check, na?
Different, sir. Different person.
Alright.
Still showing red. Plus 8 characters, lowercase letters, number and special character.
Oh, okay. Uppercase.
Uppercase. Full access. Business account. Business.
Come, sir. You come, sir. It's all because of her. Amen? Huh? Yes, sir. Very good. Thank you, Vinata. So create an empty environment, conda new environment called NGS. Conda create minus n NGS. Minus n for name. NGS, yes. So inside that we have to install next flow also. Okay? Create a new environment called NGS. Next generation sequencing. Done? Very good. Now install yes, pressing Y. So then activate the environment.
Conda activate NGS.
Yes.
Create and activate. It's still creating, just wait.
Conda activate NGS.
So can anybody tell me what is the workflow for AGS? Janata? So from the sequencing machine we start with?
So from sequencing we used to get the first Q file. So after
Madam, sir, concentrate.
yes, after that we have to do the quality check by using first QC.
Then?
And then we have to import the data get from the Linux environment. And then
Normal workflow I am saying.
yes, after quality checking, after quality checking, we have to do the
What?
trimming and all.
Trimming what? What I will trim?
After like quality checking, the bad quality it is we have to trim it
How do you know it is a bad quality?
by using the first QC paper, especially the Q score.
What is Q score?
Fret score.
What is Fret score?
It should be within 30.
What is 30? How 30?
Like error should be like less. Yeah, that is first. Yeah, error should be 1 in 1000.
So why 30? How that is coming? How 1030 is related? How 1030 is related?
Use that formula of Fret.
How you compute Fret score?
Formula was there.
Was there, will be there on the internet? Yes, it will be there. Anybody? Anybody having an idea? Okay, let's start with what is FASTQ file. What is the FASTQ file?
That is having the quality score for each and
Huh? Quality score.
yeah.
Okay. Okay, let's go one by one so that you guys understand what is what. Okay, I will share you guys one of the very important. Have you seen this video? Okay, the volume. Sound. No sound. Without sound it will be difficult. I need sound. Next generation. Your volume, no?
Hello and welcome to today's talk about next generation
Yes, sir.
sequencing, which is also known as method
It's fine. Wait.
fleet parallel or just deep sequencing. Next generation sequencing has completely revolutionized genome analysis. With next-gen sequencing, it is possible to sequence an entire human genome in just one day. We start with the sample preparation. Once we have isolated our DNA, we need to generate smaller fragments. Now all of these DNA fragments will get ligated to certain oligonucleotides. There are two types here. These include sequencing binding sites and also an additional complementary sequence which can then hybridize in the flow cell. First, our DNA double-stranded fragment is denaturated. So in theory, the whole genome should be in fragments and all of the fragments have attached two different types of binding sites on each end. All the DNA fragments are now applied on a chip, the so-called flow cell. And as you can see here, there are two types of complementary sequences, the blue ones and the red ones in our example. The DNA fragments will at this ends hybridize with the flow cell as applied to the plate. To make this easier, we just look at one fragment. The first step here on the flow cell is the DNA amplification. By polymerase chain reaction, the complementary sequence to our single-stranded DNA fragment is synthesized. This process, which we have seen here, is done simultaneously for all DNA fragments on the whole flow cell. Now the DNA is denatured, and the single strand, which is not attached to the flow cell's oligonucleotides, gets washed away. The next step is called bridge building. You may have noticed that we have two different types of oligonucleotides here, and the second type of oligo will now also hybridize to one of the sticky oligonucleotides on the plate. This process is called bridge building. The DNA is now amplified again, so a polymerase will synthesize the complementary sequence again. This whole process is called bridge amplification. The DNA is denatured, but now you can see that both fragments stick to the plate. The whole procedure will just repeat now. The DNA fragment will build a bridge again. And again we also have the bridge amplification. That whole process will be repeated for several rounds. And this here is the result. So out of one DNA fragment, we generated multiple copies by PCR. We have reverse and we have forward strands from our copy here. One of these strands here is cleaved now, and sequencing can begin. For sequencing, we just look at one copy now. As mentioned in the beginning, a primer can bind to the oligonucleotides. So this is the sequencing binding site here. Sequencing is now done with specific nucleotides. These nucleotides, in this case, are fluorescently labeled. Only one of them will fit. In this case, the T. The fluorescently labeled T will then be excited by a laser, and the fluorescent signal is obtained. This is done with a color code, so we know that in this case blue is T. In the next case, the A will join. Again, the fluorescently labeled A is excited, and we will receive a signal again, this time an A. This whole procedure is done for all of the nucleotides, one by one, and this is how we get the sequence. For simplification purposes, we just look at one of these copies. Now, notice that due to PCR we have several copies, so many light signals are obtained simultaneously. The last step now is the data analysis. So here informatic tools are required. The whole process of next-gen sequencing generated millions and billions of reads. These reads are now overlaid and compared to a reference genome. And by this overlay we can now generate the whole genome sequence. This was it. I thank you very much for watching.
So this is a very nice book. Okay, I am just sharing in the group. Which you can explore more on data visualization with R. Just take out this what you call as this paper. It's called a survey of best practices for RNA-seq data analysis. Okay, so here if you come to the figure, let us discuss this. Okay. Same. Yeah. So So you come to the first one. So there are many ways the analysis actually goes. Okay. First is the pre-analysis. So if you see, there is not only a very important concept that you need to do for quality control, but before that there are very important, very important things are there, which we call as the design of the experiment. Okay. In the first panel you see, the first is the most important thing. If you want to get out the insight from NGS, data analysis, next generation sequencing analysis, what you need to do, you need to have a very good experimental design. Okay. Then the second step is called sequencing design. So after you've done your experiment design very carefully, then you have to do for the sequencing design. Then comes the quality control. Okay. So the bioinformatics does not start from here. Bioinformatics actually starts from the beginning itself, that you have to have a very clear experimental design so that you can get best out of your experimentation or from the question that you are asking. So experiment design actually involves many types. The first thing, in what kind of library type you want to have. Means if you are going for single cell or you want to paired end. When you want to go for single cell, when you want to go for paired end. Suppose your interest is only to find transcripts, okay, or gene of interest which are differentially expressed, then you can go with single also. You don't require to go for paired end. When paired end is useful, for example, if you want to capture different isoforms of a gene, then you have to go for paired end. Okay. Then also one more thing that is very important is the sequencing length. For example, whether you want to go for a long read sequencing, okay, which will actually give you more isoform specific analysis better. Okay. Then third point, which was also kept, discussed with Akansha, where she mentioned the replicate number and the sequencing depth. Very important. If you are going for genome sequencing, then your depth is very important. Okay. And if you are going for like transcriptomic analysis and other analysis, definitely you should require replicates so that you can get more power to the study. Fourth comes to the sequencing design. For example, suppose you want to control whether you want to prepare your library with a randomized one or a spike team. Spike team is something which is called as, you know exactly that how much you are controlling for the amount of input that is there. Then in this you can actually control for a quality control, and also you can control the library size normalization. The randomization of the library prep or randomization of the sequencing run really helps us in avoiding confounding factors like experimental factors or technical factors. For example, if the sample is prepared by suppose many individuals, then that time also might actually come with to do the whether the influence of technical errors because there was many people actually doing the experiment or only a single individual. Second, we also call as batch effect, which is like if you have done your experiment in different days, that might also introduce your biasness in your data set. Then let's come to the quality control. In the quality control, you have four major sections, which is first to understand the raw reads, then you do the read alignment, then you do the quantification, then do the reproducibility. So in the raw reads, what are the things that you capture? First is the sequence qualities, okay, whichever you have sequence. Then you also see for GC content, because GC content gives you a very good idea about if there is any contamination. Then you also see the k-mer distribution, just to check that is there any biasness in the representation of the sequencing problem, or also duplicates. That those really helps you in understanding your raw data. Then second is the read alignment. So basically you want that read should be uniform and it should have a uniform GC content. Third is the quantification. Quantification, you don't want 3' biasness, or also the biotypes, or also you don't want anything which are low count data set or low count genes. Then you have a reproducibility. Reproducibility is something, for example, you did 2 biological samples or 3 biological samples, replicates. Now you know that exactly the quality or the read amount of read should be the same for those, correct? and also it will give you an idea that those samples should be close to each other than the rest of the samples. So that is what is the PCA, principal component analysis, and the correlation, and also the batch effect or batch effect if it is coming, then also you can understand. Now after you have done the pre-analysis, then comes the core analysis. What is the core analysis? Core analysis actually starts with the transcriptome profiling. Okay, then you do differential expression, then you do interpretations, for example functional profiling. So if you see in NGS analysis, this is the typically the flow that comes. If it was a genome sequence, then it will be genome profiling, then you don't do differential expression, but you will find differential variations that which will be interested. Then you do interpretation of the variations. Likewise, if you do in microbiome, you will do microbiome profiling, then you will do differential abundance of OTU, then you will do the interpretations. Then there are some advanced analysis also you do. For example, you want to do visualization, which we have actually covered, which is called as circularized, Sarcos plots and all those. And also additionally you can do genome browser, then you can use Sesame plots, there is something called splice graph and etc. for RNA-seq data. Then you have other RNA-seq type of analysis which is called as small and non-coding RNA analysis. Then you want to find out gene fusion discovery for cancer, then you require long reads or a single cell analysis. So these are also parts. So for example, if you are interested in single cell genomics, then you should first understand the above two, pre-analysis and core analysis. Then you will do the advanced analysis. Then there is also something to very important here which we call as integration. For example, let's suppose you want to understand the genetic basis of a particular disease. For example, diabetes or hypertension. So what you will do? So you will collect the blood and you will do two type of profiling. What? One, you take the genetic, that is the genome sequence you will do, or you can do a SNP based, array based sequencing where you get the SNPs. Okay. Then the second you can do is the RNA-seq or transcriptomics. So the first thing which has been given, it is EQTL. It is called expression quantitative trait loci. Okay, where you can find out what are the genetic variation that is actually controlling the expression across the disease condition and also the healthy individuals. On the second part, which is called as chromatin accessibility. Why we want to understand this? This is the part where we say that which a part of the genome or the transcriptome is having a regulatory effect, and you can find similarly with the transcription binding site. Then also you can do other things like proteomics and metabolomics, and this whole data you can do multimodal data analysis, what we call as, or also simply put it across the integration. Okay, so this review is the master place where you can actually start reading and you will understand each and everything. Okay, each and everything. We will just give you a, what we call as the flavor of one of the workflow where starting from raw data to our visualization. Okay. Okay, I hope this you will go back and read. Now next, so what are the things we have done? So we have created our NGS, correct? Now Conda activate NGS. Okay. So now let's come to our next flow. Next flow, just type in the Google next flow. Next flow, RNA. You don't need to download anything. First come to here. Yes. This is the blending thing. Are you able to see? I'm just sending this in the group also so that you have the copy. Okay. Okay. So you see this one. So this is the pipeline. Okay, I'll just open in our new tab so that we can see. Where is that? Yes. Can you see this? Very good. Okay. So first and foremost stuff. Okay. So this is like different workflows that we have. So you see this. So there are stages are there. What is this? First is pre-processing, then genome alignment and quantification. If you don't have genome, then you can also go for pseudo alignment and quantification. Then you have post-processing and final QC. Okay. And there are different aligners are there. Aligners means which will allow you to align to the genome or the transcriptome. You have aligner something like a star aligner, and you have one more aligner which is called a Salmon. Then you have star and also RSet. Then you have HiSet 2, and if you use HiSet 2, then you don't require for quantification because HiSet 2 does both. Then you have pseudo aligner, like for example Salmon, and quantification also you can do perform by Salmon itself. Okay. Just a minute. Hi Unisha. Vikas kota Vikas. What is it? Yeah, I'm sending you. Okay. Okay. So first things that we will do, there are a lot of things are there. For example, for trimming, if you see the next flow, this current next flow RNA-seq pipeline, you have many things you can do. You can concatenate the FASTQ file. You can do a subsample of the FASTQ file. Then you can also infer straightness using the Salmon workflow. Then you have FastQC, which is the software is there. Then you have UMI tools to extract the informations. To trim, you can use trim gallon. Then again, you can have to check the FastQ file whether by doing FastQC to see whether your trimming has been done appropriately. Then you have the BB splitter. Then you have short mRNA. Then you can go to different workflows. So the first workflow is to do with your pre-processing. Okay, in the box that you see. The second is the genome alignment and quantification. So if you are doing a genome alignment and quantification, you can go with all of the workflows. Like for example, you can use star, you can use HiSet 2, then you can use RSN, then you have Salmon, then you have UMI tools. Then if you are going for a Salmon, then you can directly go with all of them within Salmon itself. So one of the things that we will do, because Salmon is very easy to to start with, which is a pseudo aligner, where we will do alignment and also we will be doing the quantification of our RNA-seq data. Okay. So now let's go and try our Salmon workflow. So first thing what we need FastQC, correct? So come to your browser and as we did last time, FastQC first. FastQC Conda install. It is present in the Conda environment. So this is FastQC tools, FastQC. Yeah. So come to this FastQC. Is it? Everyone is here? FastQC. Everyone is here? Perfect. So now come to your terminal. In the end, you have activated your NGS, correct? So Conda install -G. It is the channel name. Channel name is BioConda. Then what is the software name? FastQC. Enter. Anyone who has done FastQC previously?
Yes.
Very good. So it will ask you to proceed. Yes. Press Y. So it will install all the required softwares. For example, Java is also getting installed. You see that OpenJDK, there is our Java. Dinata, you will able to bring your data now? You bring, yeah. I think that will be helpful for us. Okay. Now install the multi QC, because you will be using multi QC also. Bioconda in place of fast QC, multi QC. Just type multi QC. Bioconda multi QC.
Sir,
What?
wait, sir.
Yeah. So after you install this one, multi QC. OpenJDK is taking time, na? Yeah, because it is 340 MB. So how you will install the rest of the packages? So there are how many workflows that we had discussed in the slide? How many total? So there are different pipelines are there. How many there? 1, 2, 3, 4. 5 where?
Last.
How many lines are there? 4 pipelines total. Okay, there are 4 pipelines are there. Okay, each pipeline has different requirements. So first what we are doing, we are just using the pink one, the salmon pipeline we are running. Okay. So after we finish this one, then we will go. Multi QC is done, then I will install Salmon. I will see whether Salmon is there or not. Yes, it is there. So I can use the same thing. So see, your life is so easy when you are installing the software with Conda. Everything is like whatever it is required, whatever dependency is required, you don't have to worry much. Okay, you can install the software very easy, and that is the best benefit for using Linux system. For example, right now if you do in Windows, it might actually crash also. So now since you are doing in Linux, it is very smooth. Okay, I am done with my installation of multi QC, fast QC and Salmon. Then one more software we need, what? The trimmer. Okay. So we will go and we will install cut adapt. Cut adapt. Conda install. We have in the bio Conda only. Very nice. Cut adapt. So in the Quine, you have seen that the software that was used, it is not cut adapt. What was the software at the back end which was used in the Quine? It is trigonometric. Okay. So one more software which is called as trigonometric. You don't have to write down, because everything is, you can find this, what do you call as, softwares available in the Conda. Done. So what are the things we have installed? We have installed Fast QC, Multi QC, Cut Adapt, then Trigonometric, then Selman. Selman is for quantification and alignment, zero alignment. Okay. We have installed Trigonometric and Cut Adapt to remove the bad reads or trim the adapters. Okay. Yes. So these are the software that we have installed, and this is what it is required. And we are where right now? Till quantification. Okay. Just remember, we are where? From the pre-analysis, we are there. Only pre-analysis, where we are not doing differential expression, but only till quantification. So what are the packages or software required? Fast QC for quality check, then Multi QC for checking across samples. Suppose you have 30 or 100 samples are there. So if you do only Fast QC, you will get one report, one file, one report. But you have Multi QC, then all the reports will be combined and it will present you one single document. Okay. Then you have Cut Adapt and Trigonometric for removing your bad reads or and adapters. Okay. Very good. Then Selman is for pseudo alignment plus quantification. So now you are understanding that why we have spent so much time in Conda. Okay, after you come back from your what you call as lunch. Hey Baba, yes sir. So after you come back from your lunch, so what you will do again? You will do a revise on this one only, where we will not install Selman, but we will install other softwares, the other three pipelines. We will create specific pipelines for like the color we have. We will create NGS dash. This one is what? Violet or pink. This was the pink workflow, correct? So we will write NGS dash pink workflow. Okay? What are you doing, baby? What are you doing?
I'm searching.
Searching what? Sequence of? What is sequence?
तुम एक्टिवेट किया है? तो आई थिंक वी विल इंस्टॉल फ्रॉम द बिगिनिंग फॉर यू। आपको वो अच्छे से अपने वो नहीं किया है, इंस्टॉल नहीं किया है,
शेल
वो इंस्टॉल नहीं किया है।
एनवायरनमेंट में कौन
ये देखो उबंटू है ना, उबंटू को चला, ये नहीं चला, ठीक है ना। और उसके बाद तुम्हारा वो कहाँ है, W50 जो कॉन्डा एक्टिवेट किया, कॉन्डा एक्टिवेट है। इसमें करो, इसमें पहले कॉन्डा एक्टिवेट करो, उबंटू में आना है, कॉन्डा क्रिएट, पहले एडजस्ट क्रिएट करो।
Everyone is done? Everyone is done? Now do conda deactivate. Conda deactivate. Conda deactivate. Enter. Okay. Now create new environment. Go to your workflows. Go to your next flow workflows. Yes. Let's go with our green one. For the green one, okay? So go to your terminal and type conda create -n space ngs dash green, okay, and this is our ngs RNA sec, correct? So you will write dash RNA
Green dash RNA.
then sec SEQ, yes, enter. No space, single name, RNA sec. Very good. Then activate now. Activate the NGS green. Activate. What is the first you have to install? So come to your yes. For the green one, we have to do a installation of what? Fast QC, correct? Install fast QC. Conda install -c, channel name is what? Bioconda, then fast QC. Then you have the other one is multi QC. Then salmon.
Then what is there? What is that? Trim GIAN was there, na? Let's check whether trim GIAN is available. Trim Geller Conda. Yes, it's available. So install trim GIAN. Bioconda only, trim dash Geller. Yes. Is it done?
Yes, sir.
Very good. Then what is left? Trim GIAN is done, fast QC is done, multi QC we have done, na? Very good. Then Selman is also done. Then we require star. In the green you have star, correct? Let's do star aligner. Just type star. Bioconda. Yeah.
Something is green RNA.
Very good.
Green RNA.
Now what we have to install next?
Green RNA.
We need Selman is there? Yes? What?
Baba, where is your online environment? First create your online.
So Selman is done. Then next what? Star is also done. Next same tools. Okay. Don't do the spelling mistakes, okay? Same tools. S. Same tool is done. Then what? Picard. And there is a bad tool also there. Bad tool is there and Picard is also there. Picard. Are you able to install? Attack. After Picard, you have to install the back tools. After Picard, you have to do back tools. You give your reference this figure. Then back tool is done. Then back to weak is there. String type. String type.
फिर उसके बाद स्टार्ट फिर सेंड टू फिर फाइव सलमान के नाम से ट्रेन के लोग
R is sexy. R sexy. R sexy. हाँ पिकार्ड के बाद ये वाला है कोला से कोला Then polymer. You have in the reference the book, no?
आपको किसी को स्टार में हो स्टार
D sec you don't have to install, okay? Only till DOOP radar. That's it. Okay, DOOP radar is in the bioconductor. Okay, you don't have to install DOOP radar also. If you want to install, you can type like this. DOOP radar is a bioconductor package.
कितना सारा कुछ
Vectors what? Vectors you have to go to this out here. String type. Yes.
String type हमारे पाइथन के पास string type ordinary string type
So once this is done, I think you will finish your green pipeline. Will be done. Are you able to follow now? Where?
string type
Record.
हाँ
कहाँ तक किया सही?
बाकी
कहाँ तक किया?
Okay, after you are done, please go for lunch. Then we will come back. Okay? कोई बात नहीं, अटक गया तो फिर अच्छी बात है. That will load. Don't worry. The internet is getting. So how seamless it is to install now? So you don't require too much of trouble. So that's why we are using Conda for installing our required packages. Okay? Yes. 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 You have to first create your environment. Where is your environment? Green environment. NGS green environment. After you created, you have to activate, na? No, you have not activated. Again, do activate. First check whether it is there. Conda env list. Conda env list. Always install within. What is it saying? Yes. You should not do in the. Never do installation in base. Never ever. Okay. Your old dependencies will go here and there. Conda activate. Do Conda activate. अरे नाम तो दे दो. What name you did? What was that? Do one thing, create one more. Rather than like this, don't work. Conda activity, Conda create. Minus n. NGS green. NGS dash green. Enter. आ जाएगा बेटा, आ जाएगा. Yes. Now activate. What is saying? Conda activate. NGS green. Don't do in base. And you do in this one. Always use this. Wait, go to this one. This is the Ubuntu, okay? Now here create an environment. We'll go this. Never work in base. Create. Conda create. Now we start from beginning. Don't see this. Just see that one. Na, that you have fast QC Conda install. Type. Type then practice. Neha. Green. Enter. No. First you have to now install your first multi QC fast QC node. Conda install. Then what? The channel -C. Space -C Bioconda. The channel means Bioconda. Then the package name what? Fast QC. First install fast QC. Fast QC. Enter. Then you have to install multi QC. Then you have to install. Cut it up. No. Trim GIAN. Because we are doing the green package. Okay. Then star. Then same tools, Picard, then back tools, then string type, R set, R is small, okay? Then quant map, quality map, then do render it say bioconductor package. Till here only you do. Okay, do one thing better. Do cancel. Control C कर दो. Again do control C. हाँ, it's happening. आपका हो गया? Now you start Conda type, start typing Conda install -C install -C channel name is Bioconda and the package what you want to install fast QC. Then you have to install multi QC, then Selman. Enter first this one, then multi QC, then Selman, then trim GIAN because trim GIAN, then what was that? Star, then same tools, bed tools, Picard, bed tools, string type, then RE sec. That's it. Loop is not required. That is a bioconductor one. Okay, just install that much. That is fine. Is it okay? Everybody is done? Okay, whoever is done, please go for lunch. Okay?
नहीं आप जाइए खा लीजिए अभी वो क्या
Yes? Just like this, after you are done with this, nanopore is done.
Yes, sir.
Then after you do activation, just clone, git clone. This one you have to activate. You then you have to clone the repository for the Massimo ONT track too. Git clone and the URL. Then do CD, ONT track, CD. Yes, after the URL, just after you do this, then CD, ONT track 2. Okay, is it done? Then do ls, you will find those files. One is your config file and one is your NF. NF means next flow. Okay. And if you go to this page, first what you have to do, CD ch mode 755, okay, to the older files. For example, CD ch mode 755 star means old files. If you do a ls, you see old file have the now executable permission. 755 star space star.
Sir, in our case it's dollar.
What?
In our case dollar is coming instead of hash.
It's okay, it's fine. That's not a problem.
And then again ls.
Unless you do, you will see the difference. Before it was white, now it is green.
Yes,
Okay,
yes.
this is executable now. Okay, then what it shows is the overview of the pipeline. So if you want to use directly, you can run your next flow -c ONT, then run ONT and this one and FASTQ files. So inside the FASTQ there is a FASTQ file which is already there. Then your script directory, then result directory, then profile, Docker. I think Docker is not required.
CD on track 2.
Configuration profile to use Docker or Singular.
Where did you do? TRCK, not TRK.
We have any system which is having 10 GB of memory. So right now in the system we have 4 GB, so this will not run. Minimum processor required is 10 GB. Huh? So you can do one thing, you can follow the same tutorial for a good system, wherever it is. Just follow this, you will be able to install this. Okay? No problem. At least we came to know that this will not run in the system. Okay. Then Conda deactivate. The Conda deactivate. Let me see how much is the downloading for our genomics.
Sir, I will give you the data. It's copied I think now.
Bring in a pen drive. One file also will be good.
It's very good. No, sir, I got 3 sequences. 6.
Well, like I said. Very good.
क्या निकालना पड़ेगा
Actually,
आवड लागते
it was very better.
ভাল লাগছে না ভালো লাগছে
Yeah, but now if you see only like this is coming. Now on track was not done. So now on track is
আমনি লাগে না
तुम्ही बांधून घ्या
very nice he's speaking very nice Assamese
Sir,
yes what you want
sir, like for that one, for that one on track 2 directory we created na.
Yes.
So now we are not doing the thing.
CD
So
CD backpack
CD backpack.
yes CD type CD space dot dot enter only 2 yeah
We also do CD.
how much GB of memory you have in your system
The C drive
no memory RAM
4 GB 4 GB. It will already slow.
so I will do the demonstration and before you go for your tea please copy the data okay
मेरा तो 8 GB हुआ
so we are going to do very small what do you call as exercise to find out the differential expression between tissues so we have only 2 samples here brain gut but it's okay for them it's fine I am anyway downloading the other one more but today we will finish differential expression and pathway enrichment and all of this okay don't worry one only file go there not much that we can do anybody
Wait, anybody is having pen drive?
extra pen drive anybody
दे दो वहाँ पे भी दे दो
हाँ कंप्यूटर में तुम 2-3 को दे दो
Okay,
दे दो
दो,
बस
okay?
बस
It's around 128
GB ना ना लगे यार इफाले दिया
okay before you guys start the next session let's understand fast Q file okay can you just go to the link fast Q yeah so we can do one more thing we can just download some data directly from the FASTQ format can you go to your Conda environment install this in your Conda green NGS green Conda activate so if you have to check the environment please check Conda ENV list Honda activate NGS dash. So we can just do the NGS one because we did for this what we call a Salmon one. So Honda activate NGS. That's it. Only NGS. Now install Honda install -C Bioconda parallel fast queue dump. Fast queue dump. This one parallel fast queue dump. Just do control enter. You are installing? Yes.
Parallel dash fast queue. First queue dump dash and 6 control enter. Give him water. No space. Okay, for fast queue dump we cannot install. I think there is some issue. So what we will do? We will do Honda deactivate. Okay, Honda deactivate. Then we will create a new environment for only fast queue dump. Okay, Honda create -n fast queue dump. Is it fine? Honda create -n fast queue dump. Base में sir.
Yeah, space नहीं dash.
Base में
बेस में नहीं करना है. Environment नया environment create करना है. Because there was a conflict. Okay.
Honda create.
Then after that, again install.
अभी मुझे एक्टिव होगा
क्रिएट हो गया
Is it good? I think someone is touching. Otherwise it won't move right. Whenever I come in, the position is not good. You can see the bones behind. In the end, we are not able to focus on this end.
I'm sorry.
Yes, everything is good. Yes, everything.
खाली हम खाली से ना 9 GB
मेरा फीडबैक किसके पास है?
cái bà chưa ribbon nóng hả? Ribbon siya na om. One is
Yours. No space. No space? I mean, it's there, but it's not sufficient.
Are where are the two? Hold on here. Huh? Create DIR. Make DIR E. coli. Make DIR E. coli. MK DIR E. coli. Then go inside CD E. coli. So we will download the data from our SRA. FASTQ dump.
हो रहा है कभी एक हो गया
Everybody is downloaded.
There is a data RNA sec, so you have to copy that one.
Have you downloaded? Who has downloaded the file from the pen drive?
No.
Not yet. Okay, let me show you the.
कौन सा वाला था देना था R1
वो पूरा 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 फोल्डर को करो
RNA sec
Okay, so what data we have right now? DB R1, DB R2. Then you have gut R1 and R2. Okay, this is the data for which we have done a transcript mix, where you have done for brain, and this is for B. Correct? Where is the B person? Where is our Dinata? Where is Dinata? Let her come. Okay, first thing that what we will do, we will build a fast QC for this one. Okay, what you have to type first? Fast. Okay, first we have to go for our deactivation because we are not using this one. Deactivate. Honda activate NGS. Okay, then we have fast QC. Then, for example, we have how many are there? 1, 2, 3, 4, 5. Correct? So if you put star dot gunzip, then also you will get all the files. Okay, how you can see that? Minus L this. Okay, if you put star, it means any name but end with the dot gz. Okay, so let's run our fast QC. Fast QC and star gz, and let's try for only one file so that it becomes a little bit faster. R1 fast QC. Okay, enter. So now suppose the best part of using a command line is that you if you put star, then it will do it for you all the files. Okay, and if you have a multi-core processor, high-end processor, then also you can define if you have to do parallel, so you can get more number of cores also here. We have 4 GB of RAM, but hopefully this will work. Hopefully, let's see. First you have to copy your file. Okay, no required. Come back, cd log. First you have to go to your folder. I am in my folder, GIAN folder in the R Studio, where I kept the inside the data folder. I have given the raw folder. Inside there I have kept my data set. One file only, it is so much. I did not want to. Thank you. I can show it to my laptop. One they can submit there. Are you copying the file?
Yes, sir.
Only copy one file. I think that will be enough because it is taking a lot of time. Just to practice, just take only one file. That is fine. But I want them to practice. Just copy one file, okay? Maximum 2 you can copy. Capital D, capital O, one drive. Capital D. Then you have users with capital U. Is it done? Where is your in the admin. Okay, come to your terminal. Come to your terminal. CD now.
Zip file में Raji से बन
Then capital U. Okay, and then you will have HG. Then E na. Now try. First space any file BB. Capital B. R1. Tab. Enter. First help others. Help others. Help others. Help others. Help others.
मेरे को क्या
सबको दे दो.
तो यहाँ पे दो कॉपी कर देते हैं क्या
सब कॉपी कर लो. दो कॉपी कर लो. रीड 1 रीड 2 कर लो एटलीस्ट. रीड 1 रीड 2. रीड 1 and read 2. Read 1 and read 2 only. Okay. BB read 1 and read 2. BB R1 and BB R2 only. How you have to go? Where is your folder? First you have to go CD mount C users. Then where?
Your back.
Huh? In the E folder. Mouse E, then RNA, then MS. We have kept here. First deactivate the file. Contact deactivate.
I am copying.
Enter. Okay, then contact activate SGS.
Anyone is sitting behind?
Okay. Now do fast PC. Fast. You see the space. Capital D. R. R1. Enter. LS do LS. Yeah. Press tab. Complete file. Yeah. Now there. Yours is faster, huh? Go to the path. First tell her to go to the path. Go on to the path. First DB1, DB2. Okay, let's come for a tea and then come back. Then I think we will finish it up, and we will do the demo on top so that you are able to understand the flow. Okay, and it might take time here. My downloading is done.
almost realistic. I should say for like a office.
Coffee or lunch? Okay.
और भी है
ইমান ডাঙৰ ডাঙৰ ফাইলকেইটা একেলগে দিছে কাৰণে চাগে এটা ৰখাই দিওঁ এটা এটাকে কৰাই দিওঁ নেকি
পোষ্ট কৰি দিবা आपका
वो कर रहा है फाइल पूरा ले लिया Please study here.
She may be nervous. The activity. That was fun.
Why you miss that? I can't.
I thought you meant it.
No. Just wash with water.
You should have at least tried finished one.
No, I tried. Dangerous for me.